All right, Ninja Nerds, in this video we're going to go over triglyceride synthesis. So if you guys have already watched the video on how we actually synthesize the fatty acids, it's going to be very, very important that you guys have watched this before you watch this video. Okay, so let's go ahead and dig right into it. So if you guys remember, what are the constituents of making triglycerides? Well, one thing is we need fatty acids. So here what we have is we have 3 fatty acids, okay, but they're specifically called fatty acyl-CoAs because they have that coenzyme A on them. Now, what we're going to try to do is we're going to take these fatty acids and link them up to this chain right here, this 3-carbon foundation. And this molecule is called glycerol. So far, we need 2 things in order for this process to work. We need these 3 guys here, which are all called our fatty acyl-CoAs. Right? So these are our fatty acyl-CoAs, 1, 2, 3, because in order to make triglycerides, we need 3 fatty acids. Then we're going to have this molecule here, and this is going to be our glycerol. These are the constituents to synthesize triglycerides. And you know another word we give for synthesizing triglycerides is they also call it Lipo Genesis. So another name for it is actually specifically called Lipo Genesis. And that's what we're going to be talking about in this video. Okay. So if you guys remember, we synthesized the fatty acyl-CoAs with the malonyl-CoA acting as the precursor with those fatty acid synthetase enzymes and going through all those different steps with the acyl carrier protein and the cysteine thioend. Now that we've done that, we've synthesized our fatty acids, okay? Now that we've synthesized them, we're going to want to link these guys up. In order for us to do that, though, we need to understand something. We didn't really talk much about the glycerol. Where can that glycerol come from? Well, you know it can come from just the adipose tissue. So if you guys remember, specifically from just the adipose tissue, let's say I put over here a couple different adipocytes. So here's a couple adipocytes. And if you guys remember, we could actually break these triglycerides down, right? And one of the constituents that we can get out of that is glycerol. That's one of the constituents. We can get glycerol. And what was this from? This was from lipolysis. Where else can we get the glycerol? You know, from the glycolysis, there's a specific point in the glycolysis pathway. So let's see here I have glycolysis pathway. And a specific point in the glycolysis pathway is you form a molecule called dihydroxyacetone phosphate. And that dihydroxyacetone phosphate, if you guys remember, he was able to be converted into this glycerol molecule. So let's show how this happens. So this dihydroxyacetone phosphate can be converted into glycerol. In order for that to happen, though, I need to take and drop off some hydrides. So I need to take NADHs that I've generated and convert them into NAD positive. So what am I doing? I'm dropping off hydrides onto this dihydroxyacetone phosphate and converting carbonyls into alcohols. But another thing is, this guy has a phosphate on a specific carbon. We're going to have to put a phosphate on one of this guy's carbons too. So if we were to number this, glycerol 1, 2, 3 carbon chain, right? On the 3rd carbon of the alcohol, there should be a phosphate group coming off of it. So what should be coming off here? There should be a phosphate group coming off. So now what do we specifically call this? We call this glycerol 3-phosphate. That's what we really should call it, right? We should technically call this glycerol 3-phosphate. Before we do that, though, let's actually show that in an actual single-step reaction. Let's do that in a single-step reaction. So let's put the alcohol back on here, and then let's use a specific enzyme, technically. There's technically a specific enzyme that can do this step right here. We're going to show it as a separate step here. I'm going to take this glycerol, and I'm going to have a special enzyme. And this enzyme is called Glycerol Kinase. It's only present in the liver, right? And what this Glycerol Kinase is going to do is it's going to put a phosphate on the 3rd carbon of glycerol. So let's say I take this phosphate here and I feed it into this reaction here, okay? Now what is going to happen? Now let's draw our glycerol over again here, our 3-carbon unit here, right? With our hydrogen there, our OH here, carbon, OH, carbon, but then we have that oxygen there. Let's draw all our hydrogens. And then, now, what should be linked to this oxygen, guys? There should be a phosphate. Now, what is this molecule here called? This molecule is called glycerol 3-phosphate. So technically, if we really wanted to be particular, we could actually erase this step here from DHAP going to glycerol, because technically, if you guys know, DHAP is actually converting into glycerol 3-phosphate. So we should actually show that over here. And then again, what are we generating in this step? We're taking NADH, dropping off the hydrides to form NAD positives. Okay, so we got the dihydroxyacetone phosphate into the glycerol 3-phosphate, and that can be done straight from DHAP from glycolysis to glycerol 3-phosphate, or we can take from the adipose tissue. You know the adipose tissue has the triglycerides, and when you break them down, you break it into glycerol and fatty acids. What we can do is we can use the glycerol here, and I have an enzyme called Glycerol Kinase. And Glycerol Kinase can convert glycerol into glycerol 3-phosphate. Okay. Now, the next thing that we have to do here, then we have to take this glycerol 3-phosphate and combine it with each one of these fatty acyl-CoAs. So now what I'm going to do is I'm going to draw the glycerol in a large view here. So now we're going to take this glycerol step by step here. So let's say that here we have our carbon. I'm going to draw it like this now. And I'm going to put my OHs right here. And again, on the third carbon, I shouldn't have an OH. I should have a phosphate group coming off. Let's draw all the hydrogens coming off of this puppy. Okay, there's our hydrogen there. There's our hydrogen there. Okay. And again, let's number it just so we know exactly where we are. 1 carbon, 2 carbon, 3 carbon. But you would know that because on the third carbon there is a phosphate group. Okay. Now, what's the first step? The first step is I'm going to number each one of these fatty acids. I'm going to number this fatty acyl-CoA number 1. I'm going to number this fatty acyl-CoA number 2. And this fatty acyl-CoA number 3, because we're going to add each one of these on in sequence, because it's very particular in the way we have to do this. So the first thing we're going to do is we're going to add this fatty acyl-CoA onto the number 1 carbon. So now I'm going to take this guy here, and I'm going to add it onto that 1 carbon. So let's show that. So this guy right here is going to come in, and it's going to be added onto that 1 carbon. Okay? Then, if you want to be particular, this guy here would be added onto the 2nd carbon. And this guy right here would be added onto the 3rd carbon. But there have to be a specific sequence of actions of how this is happening. So now what we're going to do is we're going to show this in a sequence of steps of how it's going. Okay, so now let's have our carbon group here. Hydrogen, 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 hydrogen again. And then let's have our OH here, OH, and then again our oxygen with the phosphate group. Okay. So let's take that first fatty acyl-CoA that we're going to call fatty acyl-CoA number 1. So we're not going to show that whole structure. We're just going to say specifically this one is going to be fatty acyl-CoA number 1. I'm going to add this guy into this guy here. So I'm going to take and fuse these two guys together. But when this guy reacts with this guy, we're going to specifically, again, let's get our carbon straight here again. This is carbon 1, carbon 2, and carbon 3. This fatty acyl-CoA number 1 and this glycerol is going to be acted on by a special enzyme. And this enzyme is called acyl-transferase. So it's not hard to remember because this enzyme is transferring a fatty acyl-CoA on here. And what are you going to lose? You're going to lose a coenzyme A out of this reaction. So you'll lose a CoA. But this fatty acyl-CoA number 1 is going to be added onto the number 1 carbon. So now let's show that. So boom, boom. And again, I'm going to have my hydrogen here, still going to have my phosphate group there. That hasn't changed. But now, look what's going to be different now. On the first carbon, I'm going to have my oxygen there. But now, what am I going to have? I'm going to have this fatty acyl-CoA added to it, but without the CoA. So let's draw that. So now what am I going to have? I'm going to have carbon, double bond oxygen, and this is going to have a CH2, 14 carbons like that with a CH3. What do I call this molecule? This molecule is called phosphatidic acid. So this first molecule that we just formed is called lysophosphatidic acid. Okay, so that's the first one that we formed. We formed our lysophosphatidic acid. Okay, Now that we formed our lysophosphatidic acid, let's do the second step. So now in the second step, it's just going to be the same thing. So look here in the second step. In the second step, what am I going to be adding here? I'm going to be adding the second fatty acyl-CoA. I'm going to add this guy. So I'm going to add fatty acyl-CoA number 2. So these two guys are going to react. But again, the enzyme that's catalyzing this step here is still going to be a acyltransferase enzyme. So what is the enzyme triggering this reaction to occur? Again, the enzyme is going to be specifically a acyltransferase enzyme. Okay. And again, what are you going to lose out of this step here? You'll lose a coenzyme A. So out of this will come a coenzyme A. Now let's draw this new molecule right here. So now we're going to have our carbons with all the hydrogens attached here again. But now, if I redraw this guy here, we have our first the fatty acid added on here. And then what are we going to have? We're going to have another fatty acid added on. So now let's add this next fatty acid. So how does this reaction occur? I'm going to add in this guy here. But here's the key thing. What do you notice the difference between this guy and this guy? This guy has double bonds. So he's an unsaturated fatty acid. This is a saturated fatty acid. Saturated just means that it has no double bonds. Unsaturated means it has a double bond. If it's monounsaturated, it has one double bond. If it's polyunsaturated, it could have many double bonds. The critical thing is that the second fatty acid added has to be unsaturated. Okay, so now we're going to add on this second fatty acid, but it has to be unsaturated. So now let's add that on. So now we're going to have, again, the carbonyl. And then what does it have? It has a CH2. Then it's going to have a CH2, but 7 of them like that. Then you're going to have a carbon, which has a double bond with another guy. And then what? Then you're going to have a CH3. This is our next molecule. What is this guy here called? This guy is specifically, let's draw our oxygen here with the phosphate group. Need to be consistent with our molecule here. This should have a phosphate. This should still have a phosphate. And again, this was called lysophosphatidic acid. This is just going to be called phosphatidic acid. So this molecule is called phosphatidic acid. I keep spelling my things wrong. Phosphatidic acid. Okay, so we got phosphatidic acid comes after, and then there was lysophosphatidic acid, and before that we just had the glycerol, right, as our basic unit, glycerol 3-phosphate. Last step. Now what we're going to do, well, actually there's 2 more steps, but it's going to be simple. We're going to combine these 2 steps together. If I combine 2 steps together, I'm going to do the same thing I did in the first 2 steps. So again, this was step 1, where I added fatty acyl-CoA number 1. This was step 2, where I added fatty acyl-CoA number 2. In the third step, I'm going to do 2 things. I'm going to add in, what you guys already know, I'm going to add in my fatty acyl-CoA number 3. I'm going to add this one in too. And what am I going to lose out of the process of this? I will lose a coenzyme A. So I'll lose a coenzyme A out of this process. Coenzyme A, but here's the kicker. Look, can I add that fatty acyl-CoA onto this end? No, because there's a phosphate there. So I can't get that, I have to get that phosphate off first. So what do I have to do before I add the fatty acyl-CoA? I have to bring in a special enzyme. What is this special enzyme? The special enzyme is called a phosphatase. Phosphatase. And what's this phosphatase going to do? So technically it's a phosphatidic phosphatase. It's going to come in and it's going to cut that bond right there. It's going to cut this bond here and it's going to release out a phosphate. So what enzyme is going to do this? A phosphatidic phosphatase will cleave that phosphate off of the 3rd carbon. So what am I going to release out of this reaction? I'm also going to release a phosphate. Then I'm going to add the fatty acyl-CoA number 3 onto the 3rd carbon there with the oxygen, right? So now let's draw our finished triglyceride product. So again, let's draw it here at an angle. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, another hydrogen. And again, let me draw all my oxygens here. And now let's draw our finished product. The first one had specifically a carbonyl with a CH2, 14 of them like that, and then a CH3. The second carbon had specifically what? It had a carbonyl with a CH2, then it had CH2, 7 of them like that, then it had a double bond, and then a CH3. And the last one specifically had what? This one is going to be exactly the same as the first one. So what are we going to have here? Carbon, double bond oxygen, CH2, 14 of them like that, and then CH3. What is this molecule here called? This is called a triglyceride. But sometimes they refer to triglycerides, they give them a different name. They call it a triacylglycerol. Okay, sometimes you might even see it referred to as a triacylglycerol. So be prepared to hear either one of those terms, because a triacyl, this is a fatty acyl, this is a fatty acyl, this is a fatty acyl. So we're adding on fatty acyl groups. So you can call this a triacylglycerol or just a triglyceride, because you know another way that we can name these? Technically, this one is specifically a monoglyceride or a monoacylglycerol. This one is technically considered to be what? A diacylglycerol, and this one is a triacylglycerol. So I can also kind of name these different names too if I want to. I could technically call, I could call this one a DAG, which stands for diacylglycerol for this molecule right here. And I could technically call this one a MAG, a monoacylglycerol. And again, this one is a triacylglycerol. All right, so this covers the triglyceride synthesis. Now the next question is, and we're going to cover this when we link this to cholesterol metabolism, is what can I do with these triglycerides? Well, these triglycerides can actually be packaged into special different types of lipoprotein molecules. Those lipoprotein molecules that we're going to package them in is going to be called very low density lipoproteins. So what we're going to talk about in another video is specifically me taking this triglyceride and packaging them into special structures. And again, what is these special structures? Let's say I draw it here, and I can put this whole triglyceride, which I'm going to just draw now in a different way, but meaning the same concept here. So now let's have a green fatty acid, we'll have a blue fatty acid, and we'll have a black fatty acid. And this is going to be in a specific type of lipoprotein molecule with specific types of apoproteins, which we'll talk about. And this is going to be called a VLDL. And where can this VLDL go? It can take and transport this triglycerides and a tiny little bit of cholesterol to adipose tissue, where it can be deposited into the adipose tissue and stored as triglycerides. So again, where could this VLDL go? This VLDL could come over here. Where? It could come over here and do what? It could be transported through the bloodstream and then deposit those triglycerides into the adipose tissue. So what is it going to throw in here? It's going to throw into the adipose tissue. I'm just going to draw it here in a general way, guys. It's actually going to get broken down by lipoprotein lipase. But what's going to happen is it's going to get taken into these adipocytes and then resynthesized back into triglycerides, where they can be stored. Okay? So just a really quick recap. Again, we need glycerol as our basic unit, which can come from glycolysis, from the DHAP, or it can come from the actual breakdown process from the adipose tissue like lipolysis. And again, this is also can happen a little bit in the liver, but not too much. But then the glycerol, what can happen? Only in the liver you have this Glycerol Kinase enzyme, which does what? Converts the glycerol to glycerol 3-phosphate, which is the basic unit for what? For these fatty acyl-CoAs to be added. And what did I do? I added each fatty acyl-CoA in a sequence. 1, 2, and 3. And again, what would be the enzyme I didn't mention here, but it's the same enzyme that we mentioned in all of these, acyltransferase in the third step again. But all you're doing is you're adding these fatty acyl-CoAs in sequence with an acyltransferase present. Okay, converting it into what? So taking glycerol, adding a fatty acyl-CoA and going into lysophosphatidic acid or a monoacylglycerol, taking that lysophosphatidic acid or the monoacylglycerol and taking another fatty acyl-CoA, adding that in with the acyltransferase and forming phosphatidic acid or a diacylglycerol, taking that phosphatidic acid and diacylglycerol, doing what? Getting rid of the phosphate with the phosphatase enzyme, and then adding another fatty acyl-CoA with another what? What is that enzyme again in this step? Acyltransferase. And again, what do you form? You form a triacylglycerol. What can we do with these triacylglycerols? Specifically, you're going to take these triacylglycerols in the liver and package them into special lipoprotein molecules called very low density lipoproteins. And where do they take that triglycerides? They take them to the adipose tissue. They have a specific protein on their membrane that recognizes a specific enzyme here called lipoprotein lipase, which cuts the triglycerides and releases them into the adipose, where they'll be resynthesized back into triglycerides and stored when we need to utilize them again. All right, engineers, so in this video we covered triglyceride synthesis. In another video, we'll kind of talk a little bit more about these lipoproteins when we discuss specifically the cholesterol metabolism. All right, engineers, I hope it all made sense. I hope you guys enjoyed it. If you did, hit the like button, subscribe, and leave a comment down in the comment section. Till next time, engineers.